Friends, today we will study about micropropagation, or we can say clonal propagation. Details we will study. Plants can be propagated bisexually or asexually. Clonal propagation refers to the process of asexual reproduction by multiplication of genetically identical copies of individual plants. The term clone is used to represent a plant population derived from a single individual by asexual reproduction. Asexual reproduction through multiplication of vegetative plants is the only method for the in vitro propagation of certain plants as they do not produce viable seeds. Example: banana, grapes, fig, chrysanthemum, like this. Clonal propagation has been successfully applied for the propagation of apple, potato, tuberous, and several ornamental plants. Now let's know about the advantages of vegetative propagation. Asexual propagation of plants has certain advantages over sexual propagation. First point, faster multiplication. Large number of plants can be produced from a single individual in a short period. Possible to produce genetically identical plants. Sexually derived sterile hybrids can be propagated. Seeds raised plants pass through an undesirable juvenile phase, which is avoided in asexual propagation. Gene banks can be more easily established by clonally propagated plants. Now let's know about in vitro clonal propagation. In the in vivo clonal propagation of plants is tedious, expensive, and frequently unsuccessful. In vitro clonal propagation through tissue culture is referred to as micropropagation. Use of tissue culture technique for micropropagation was first started by the scientist named Morel in 1960 for propagation of orchids and is now applied to several plants. Micropropagation is a hardy technique for rapid multiplication of plants. Technique of let's know about technique of micropropagation. Micropropagation is a complicated process and mainly involves in three stages. 1, 2, 3. Some authors add two more stages like stage 0 and stage 4 for more comprehensive representation of micropropagation. Now let's know brief about the stages. Stage 0 we'll start from stage 0. Stage 0, this is the initial step in micropropagation and involves the selection and growth of stock plants for about 3 months under controlled conditions. Stage 1. In this stage, the initiation and establishment of culture in a suitable medium is achieved. Selection of appropriate explants is important. The most commonly used explants are organs, shoot tips, and axillary buds. The chosen explant is surface sterilized and washed before use. Now let's know about stage 2. It is in this stage the major activity of micropropagation occurs in a defined culture medium. Stage 2 mainly involves multiplication of shoots or rapid embryo formation from the explant. Stage 3. This stage involves the transfer of shoots to a medium for rapid development into shoots. Sometimes shoots are directly planted in soil to develop roots. In vitro rooting of shoots is preferred while simultaneously handling a large number of species. Now let's know about stage 4. This stage involves the establishment of plantlets in soil. This is done by transferring the plantlets of stage 3 from the laboratory to the environment of greenhouse. For some plant species, stage 3 is skipped and unrooted stage 2 shoots are planted in pots or in suitable compost mixture. The different stages till now we discussed for micropropagation are particularly useful for comparison between two or more plant systems. Besides better understanding, it may, however, be noted that not all plant species need to be propagated in vitro through all these five stages we discussed. Micropropagation mostly involves in vitro clonal propagation by two approaches. First approach is multiplication by auxiliary buds or apical shoots, and second is multiplication by advantageous shoots. Besides these two approaches, the plant regeneration processes, namely organogenesis and somatic embryogenesis, may also be treated as micropropagation. Hence, they are also now we will discuss. First is organogenesis. The formation of individual organs such as shoots, roots directly from an explant or from the callus or cell culture induced from the explant. Next is somatic embryogenesis, the regeneration of embryos from somatic cells, tissues or organs. Multiplication by auxiliary buds and apical shoots. Quiescent or actively dividing meristems are present at the axillary and apical shoots. The axillary buds located in the axils of leaves are capable of developing into shoots. In the in vivo state, however, only a limited number of axillary meristems can form shoots. By means of induced in vitro multiplication in micropropagation, it is possible to develop plants from meristem and shoot tip cultures and from bud cultures. Meristem and shoot tip cultures. Let's know about the meristem and shoot tip culture. Apical meristem is a dome of tissue located at the extreme tip of a shoot. The apical meristem, along with the young leaf primordia, constitutes the shoot apex. For the development of disease-free plants, meristem tips should be cultured. Meristems or shoot tip is isolated from a stem by a V-shaped cut. The size should be 0.2 to 0.5 mm of the tip is critical for culture. In general, the larger the explant, the better are the chances of culture survival. For good results of micropropagation, explants should be taken from the actively growing shoot tips, and the ideal timing is at the end of the plant's dormancy period. The most widely used media for meristem culture are MS medium and whites medium. Now let's know about this stage. In first stage, in stage 1, the culture of meristem is established. Addition of growth regulators, namely cytokinins, kinetin BA, and auxin NAA or IBA, will support the growth and development. In stage 2, in stage 2, what happens? Shoot development along with axillary shoot proliferation occurs. High levels of cytokinins are required for this purpose. And let's know about the stage 3. Stage 3 is associated with the rooting of shoots and further growth of platelets. The shoot formation, root formation is facilitated by low cytokinin and high oxygen concentration. This is opposite to shoot formation since a high level of cytokinins is required. Consequently, stage 2 medium and stage 3 medium should be different in composition. The optimal temperature for culture is in the range of 20 to 28 degrees centigrade. Lower light intensity is more appropriate for good micropropagation. Bird cultures. The plant buds possess quiescent or active meristems depending on the physiological state of the plant. The two types of birds cultures are used: single node culture and auxiliary bird culture. Single node culture. This is a natural method for vegetative propagation of plants both in vivo and in vitro conditions. The bud found in the axil of leaf is comparable to the stem tip for its ability in micropropagation. A bud along with a piece of stem is isolated and cultured to develop into a plantlet. Closed buds are used to reduce the chances of infections. Let's know about the auxiliary bud culture. In this method, a shoot tip along with auxiliary bud is isolated. The cultures are carried out with high cytokinin concentrations. As a result of this, apical dominance stops and auxiliary buds develop. For a good auxiliary bud culture, the cytokinin or auxin ratio is around 10 is to 1. This is, however, variable and depends on the nature of the plant species and developmental stage of the explant used. In general, juvenile explants require less cytokinin compared to adult explants. Sometimes the presence of apical meristem may interfere with axillary shoot development. In such a case, it has to be removed. Now let's know about the multiplication by adventitious shoots. The stem and leaf structures that are naturally formed on plant tissues located in sites other than the normal leaf axial regions are regarded as advantageous to shoots. There are many advantageous shoots which include stems, bulbs, tubers, and rhizomes. The advantageous shoots are useful for in vivo and in vitro clonal propagation. The meristematic regions of advantageous shoots can be induced in a suitable medium to regenerate to plants. Now let's know about the factors affecting micropropagation. There are many factors. First is genotype of the plant. Selection of the right genotype of the plant species by screening is necessary for improved micropropagation. In general, plants with vigorous germination and branching capacity are more suitable for micropropagation. Physiological status of the explant. Explants from more recently produced parts of plants are more effective than those from older regions. Good knowledge of donor plants' natural propagation process with special reference to growth stage and seasonal influence will be useful in selecting explants. Now, let's know about the culture media. The standard plant tissue culture media are suitable for micropropagation during stage 1 and stage 2. However, for stage 3, certain modifications are required. Addition of growth regulators like auxin and cytokinins and alterations in mineral composition are required. This is largely dependent on the type of culture, meristem or bud, etc. Now come to culture environment. Light. What do we need for culture environment? First is light. Photosynthetic pigment in culture tissues do absorb light and thus influence micropropagation. The quality of light is also known to influence in vitro growth of shoots. Example: blue light induced bud formation in tobacco shoots. Variations in diurnal Diurnal illumination also influenced micropropagation. In general, an illumination of 16-hour day and 8 hours night is satisfactory for shoot proliferation. Now come to temperature. Majority of the culture for micropropagation requires an optimal temperature around 25 degrees centigrade. There are, however, some exceptions. Example, begonia cross Chimentha hybrid tissue grow at a low temperature around 18 degrees centigrade. Now come to composition of gas phase. The constitution of the gas phase in the culture vessels also influences micropropagation on organized growth. Growth of cells is generally promoted by ethylene, oxygen, carbon dioxide, ethanol, and acetaldehyde. Now come to factors affecting in vitro rooting. A general description of the factors affecting micropropagation we till now discussed. For efficient for efficient in vitro rooting during micropropagation, low concentration of salts, reduction of half to one half to one quarter from the original is advantageous. Induction of roots is also promoted by the presence of suitable oxygen. Organogenesis. Let's know about organogenesis. Organogenesis is the process of morphogenesis involving the formation of plant organs, that is shoots, roots, flowers, buds from the explant or cultured plant tissues. It is of two types: direct organogenesis and indirect organogenesis. Let's know about direct organogenesis. Tissues from leaves, stems, roots, and inflorescence can be directly cultured to produce plant organs. In direct organogenesis, the tissue undergo morphogenesis without going through a callus or suspension cell culture stage. The term direct adventitious organ formation is also used for direct organogenesis. Induction of adventitious shoot formation directly on roots, leaves, and various other organs of intact plants is a widely used method for plant propagation. This approach is particularly useful for herbaceous species. For appropriate organogenesis in culture system, exogenous addition of growth regulators, auxin and cytokinin is required. The concentration of the promoting substance depends on the age and nature of the explant besides the growth condition. Now come to indirect organogenesis. When the organogenesis occurs through callus or suspension cell culture formation, it is regarded as indirect organogenesis. Callus growth can be established from many plants like leaves, roots, cotyledons, stems, flowers, petals, etc. for subsequent organogenesis. The explants for good organogenesis should be mitotically active immature tissues. In general, the bigger the explant, the better the chances for obtaining viable callus or cell suspension cultures. It is advantageous to select meristematic tissues for efficient indirect organogenesis. This is because their growth rate and survival rate are much better. For indirect organogenesis, the cultures may be grown in liquid medium or solid medium. Many culture media like MS, B5, whites, etc. can be used in organogenesis. The concentration of growth regulators in the medium is critical for organogenesis. By varying the concentrations of auxin and cytokinins, in vitro organogenesis can be manipulated. Low auxin and low cytokinin concentration will induce callus formation. Low auxin and high cytokinin concentration will promote shoot organogenesis from callus. High auxin and low cytokinin concentration will induce root formation. Now come to somatic embryogenesis. The process of regeneration of embryos from somatic cells, tissues or organs is regarded as somatic embryogenesis. Somatic embryogenesis may result in non-zygotic embryos or somatic embryos, parthenogenetic embryos, and androgenic embryos. In general usage, when the term somatic embryos is used, it implies that it is formed from somatic tissues under in vitro conditions. Somatic embryos are structurally similar to zygotic embryos, and they are they can be excised from the parent tissues and induced to germinate the tissue culture media. Development of somatic embryos can be done in plant cultures using somatic cells, particularly epidermis, parenchymatous cells of petioles, or secondary root phloem. Somatic embryos arise from single cells located within the clusters of meristematic cells in the callus or cell suspension. First, a pro embryo is formed when which then develops into an embryo and finally a plant. Two routes of somatic embryogenesis are known directly and indirectly. Direct somatic embryogenesis. Let's know about direct somatic embryogenesis. When the somatic embryos develop directly on the excised plant or explant without undergoing callus formation, it is referred to as direct somatic embryogenesis. This is possible due to the presence of pre-embryonic determined cells, that is PEDC, found in certain tissues of plants. The characteristic features of direct somatic embryogenesis is avoiding the possibility of inducing somaclonal variations in the propagated plants. Now let's know about the indirect somatic embryogenesis. In indirect embryogenesis, the cells from explant are made to proliferate and from callus from which cell suspension cultures can be raised. Certain cells referred to as induced embryogenic determined cells from the cell suspension can form somatic embryos. Embryogenesis is made possible by the presence of growth regulators and under suitable environmental conditions. Somatic embryogenesis can be carried on a wide range of media. The addition of the amino acid L-glutamine promotes embryogenesis. The presence of oxygen such as 2,4-dichlorophenoxyacetic acid is essential for embryo initiation. On a low oxygen or no oxygen medium, the embryogenic clumps develop into mature embryos. Indirect somatic embryogenesis is commercially very attractive since a large number of embryos can be generated in a small volume of culture medium. The somatic embryos so formed are synchronous and with good regeneration capability. Now let's know about the artificial seeds from somatic embryos. Artificial seeds can be made by encapsulation of somatic embryos. The embryos coated with sodium alginate and nutrient solution are dipped in calcium chloride solution. The calcium ion induces rapid cross-linking of sodium alginate to produce small gel beads, each containing an encapsulated embryo. These artificial seeds can be maintained in a viable stage till they are planted. Now come to applications of micropropagation. Micropropagation has become a suitable alternative to conventional methods of vegetative propagation of plants. There are several advantages of micropropagation. High rate of plant propagation. Through micropropagation, a large number of plants can be grown from a piece of plant tissue within a short period. Another advantage is that micropropagation can be carried out throughout the year, irrespective of the seasonal variations. Further, for many plants that are highly resistant to conventional propagation, micropropagation is the suitable alternative. The small sized propagules obtained in micropropagation can be easily stored for many years germplasm storage and transported across international boundaries. Now let's know about the production of disease-free plants. It is possible to produce disease-free plants through micropropagation. Meristem tip cultures are generally employed to develop pathogen-free plants. In fact, micropropagation is successfully used for the production of virus-free plants with plants of sweet potato, cassava, and yam. Now let's know about the production of seeds in some crops. Micropropagation through auxiliary bud proliferation method is suitable for seed production in some plants. This is required in certain plants where the limitation for seed production is a high degree of genetic conservation. Example, cauliflower, onion. Now let's know about the cost effective process. Micropropagation requires minimum growing space. Thus, millions of plant species can be maintained inside culture while in a small room in a nursery. The production cost is relatively low, particularly in developing countries where the manpower and labor charges are low. Now let's know about the automated micropropagation. It has now become possible to automate micropropagation at various stages. In fact, bioreactors have been set up for large scale multiplication of shoots and bulbs. Some workers employ robots for micropropagation, and this further reduces production of cost plants. Now let's know about the disadvantages of micropropagation. First is the contamination of cultures. During the culture of micropropagation, several slow growing microorganisms contaminate and grow in cultures. The microbial infection can be controlled by addition of antibiotics or fungicides. However, this will adversely influence propagation of plants. Now come to brewing of medium. Micropropagation of certain plants, example woody perennials, is often associated with accumulation of growth inhibitory substances in the medium. Chemically, these substances are phenolic compounds which can turn the medium into dark color. Phenolic compounds are toxic and can inhibit the growth of tissues. Brewing of the medium can be prevented by the addition of ascorbic acid or citric acid or polyvinyl pyrrolidone to the medium. Any questions regarding this you can ask me in the comment box. Please subscribe my channel. Thank you.